Hello, everybody. Hope you are still healthy. Today, Monika and I, we will guide you through translational toxicology in drug development and in discovery. This is the drug discovery development process, the workflow, very complicated. I don't want to go into the details. It's a very regulated process, very complex. And it's also accompanied by the regulators. This is in fact the work which has to be done, the rocky road to success, where from 10,000 compounds, one compound is selected, which becomes a drug. That happen at the different levels during the drug discovery phase in order to find an efficacious drug. And then at the development phase and in the early or late discovery phases, where the drug safety aspect is addressed and where it's also already influenced, and the attrition is influenced heavily then by the safety. Now, drug-induced liver injury is one of the major causes for drug attrition. There are some companies, they have a low rate, others have a higher rate, it's depending from their chemistry. The first step in this attrition it's a heavy step when it comes into the treatment of animals. When animals are treated, the regulatory species, non-rodent and rodent species, are first treated by drug candidates. Then some companies, they have a dropout of 40 or even higher rates. If a compound makes it into the clinical phase, then it's clean. There is a safety margin in terms of safety. There's a suitable window between efficacy and safety. And even there, some compounds show some toxicity under the clinical testing phases. And then even if a compound makes it through the testing phases, there are some drops out because of idiosyncratic reactions after reaching the market. So, and one of the important pieces, or which is essential to understand, is what is the underlying mechanism. There are certainly some mechanisms which one can explore, but in the end, the individual drug has to be evaluated for its individual mechanisms, and we're talking here about the specific adverse outcome pathways of these compounds. So what makes the drug development so challenging? There's an interesting paper was coming out in 2018. It was published by the EMA. And it documents that over a long period of time, about 90% of the IND entering compounds, which entering the clinical phases, they failed. So there are multiple reasons for that, but significant reasons is the lack of efficacy and also the lack of safety. So this already indicates that we have here a tremendous need for improved translation to man. And in the last 2 years, there's some new spirit coming into the system by the discovery that the 3D spheroidal model, that they have some outstanding features compared to the 2D models. And this is a very promising tool which could make then also the translation to the clinic. So it has been shown in some cases that these 3D models can contribute to better safety evaluations, but also in terms of drug discovery, that was very helpful. So what are these mysterious 3D models? These are multicellular spheroidal models, which is the smallest functional unit of the liver. It is also a co-culture of parenchymal cells, endothelial cells, Kupfer cells, and also stellate cells. For some particular reasons, stellate cells are also to be implemented, and it shows some improved behavior and also improved features. This comes from the production process of these spheroids. They do not have an artificial matrices. They have cell-cell adhesion, and this is giving a completely different type of outcome. And you see on the right side 2 heat maps. On the left side, this is native liver together with 3D cultures cultured over a period of time. And on the right hand, you see 2D cultures also over different cultured of time. And you see that these heat maps, in terms of the proteomic profiles, completely are distinct and they differ. So this indicates that we have here two complete conditions. 2D is completely different to 3D. One particular feature of these spheroids makes them so attractive. It's the longevity of the cultures and the possibility to do here some multiple treatment culturing cell cultures over a period of time after daily treatment and according to the particular clinical setting which you are aiming for a drug compound. That gives completely new results in terms of the treatment schedule, and the outcome is also fascinating. So these models, they can be maintained, but they can also be cultured in a very robust and reproducible way. And the data which we obtained in the meantime, since we have that model, is fascinating in terms of the reproducibility and robustness. And we are currently ongoing to make the transition from 96 well to 384 well plates, where these special type of investigations can be done under high throughput conditions. So just for your imagination, there is one micro tissue in one well in a 384 well plate format, and there's a supernatant of 70 microliter. And this 70 microliter enables you a whole range of biochemical biomarker, which you can see now in the following slide. So there are several investigative targets for the DILI, which we have really to look at it, in particular when we constructing adverse outcome pathways, which are specific for a new compound. We do not know where a compound goes, what are the mechanisms, and there we have here a very broad frame of investigative targets, but also some biomarker at the different levels of complexity. It starts from histopathology, to morphology, where we can look on high content analysis and particular biochemical analysis, which are important to prove here also then the involvement of a particular pathway in the mechanism of action. And then of course a whole range of omics which can be done on this one single spheroidal approach. These are the application in translational toxicology. And on the left side, you see the classical approach, which is the DILI screening approach. This is a predictive toxicology part. It's based on data science. It investigates compounds, a large number of compounds, and the publication which is below this predictive toxicology box is a publication showing that when 108 clinical compounds were tested in the spheroidal model, the outcome was that these compounds' activity in terms of induction of DILI, their clinical DILI induction correlated to the cytotoxicity in the system. So this gives a good correlation to the clinical annotation. This approach is a statistical approach, and in the end you will get some specificity and sensitivity. So In this case, the sensitivity was as double as high as it was obtained for the 2D model. So I don't want to go into the details. I would like to focus in this talk here about causality assays, about the causality approach, which is a completely different approach from the statistical predictive toxicology approach. It is evidence-based, it depends on individual cases, and it investigates the sequence of events which a compound is inducing, and it also validates the pathway, so that when you have a sequence of events, which is not necessarily then telling you that this is a mechanism of action, many of these events can happen in parallel, but by the validation of the individual pathway, it allows you to say, this is my pathway of interest where I believe that, that my DILI toxicity is coming from the impairment of this mechanism. So in order to understand a DILI pathway of a new compound, and also to explain a new mechanism of action for DILI, it's important to see in the literature and also to summarize what in fact has been done by other DILI compounds, what is in common of those compounds, where do they differentiate, and I think this is an excellent framework to start by as an investigative toxicologist and farmer when you have such an issue. So it starts, in fact, by the molecular initiation. You have a chemical compound that can be a drug candidate, it can be a failed drug, which you have to investigate in an in vitro model, and the uptake and the absorption, distribution, excretion, and the drug metabolism plays an important role. So that's what investigative toxicologists always are doing first when they're trying to hunt after an ideally case coming from preclinical or in the clinical phases. So these molecular events are triggering cellular events, which we know from these many existing cases, which can be at the subcellular levels, which can be at the level of the mitochondria, the endoplasmatic reticulum. It can be at the level of some particular mechanism of pathways, which Monika will show one pathway, the glutathione pathway, which is an important one for the modulation of the cellular responses. And at the organ level, DILI, of course, is expressed at the levels where you really can see it when you treat animals and when you take out the liver and you're looking on the histological slides, or you're measuring some clean path parameters like cholestasis, which is a bile acid homeostasis disturbance. The steatose is accumulation of fat and fatty cumulative processes. The fibrosis, where hepatocytes are killed, where it starts to generate some fibrotic tissue, collagen-like tissues. The hepatitis, which is also unique for the liver, since there is some hepatitis inflammatory processes coming from the inflammatory cells like Kupffer cells, or for resident macrophages or cells from the immune system just infiltrating into the liver, or just creating this inflammatory environment. So necrosis and apoptosis can also happen in other tissues, but when you have it in the liver necrosis, then you see that by the release of ALT, which reaches into the periphery, and that indicates here some liver necrosis and liver damage induced by broken membranes. So at the level of the organism, you see it then in the clinics, mostly where you have individual cases causing some acute liver failure. That can be measured by Haas law cases, by bilirubin and ALT, but also you see then when a compound is on the market, you see then when several risk factors come together, and then you observe this idiosyncratic reaction. So genetic at the population level, there's also some influence from the genetics. There's some epigenetics which plays a role. There are some diseases, underlying diseases, and also sex and obesity plays a role here. So what are we doing when we want to investigate a particular mechanism of DILI? And then there is a roadmap, and I constructed here, let me say, not a roadmap. You see here the gobblestones from this roadmap, and you see the different components, and this is how it looks in the beginning when you exploring an investigative case where you have a DILI case. So, and I'm trying to figure out here 3 of the major pathways which are currently investigated when you have such a DILI case, and of course drug metabolism plays the first role, because it's telling you about the fate of a compound in an organism, and then you have the possibility to go for the oxidative stress mitochondrial damage axis, which then gives you also some indication on steatosis and also necrotic part. Here then you have the biliary efflux axis, where the drug metabolite or the parent drug can also cause here direct efflux disturbances by inhibiting efflux or uptake transporters, which then could lead, in the case of cumulated bile acid cellular contents, to necrosis. So this is a mechanism which I was recently also involved in publishing that when I was at Novartis. And then we have a very specific one, which is the one to explain DILI. That starts by the drug metabolism. It starts by the reactive metabolite formation. This is a process where a reactive metabolite is covalently binding to protein, and that gives some adducts which then stimulate the immune reaction by hapten formation and activating then T cells and secretion of cytokines. So this gives a lot of inflammation that can end up in fibrosis, but it can also end up in the necrosis. And this is one of the most prominent pathways which will be investigated in terms of DILI. So how to investigate that? It starts by the study background. It starts by histopathology, where the scene is, the stage is provided by giving a clear picture on the histopathology. That helps to create an hypothesis, and that helps then also to select a sequence, to select a set of biomarker which can be measured after different time points, and which allow to formulate here a sequence of events, which then after that can be validated for their causal link in the pathway. So that gives the adverse outcome pathway of such a mechanism. So how to do that? That's now on this slide. It starts by a signal that, in this case, what I'm showing today is the signal of the cytotoxicity, which has been proven to be involved in predicting liver DILI, and when you're approaching that in a statistical way. And this signal can be modified by modulators, so by agonists and antagonists. So this is what you know from classical pharmacology, that a signal, in terms of its validation, where it's coming from, and whether it's relevant or not relevant, has to be challenged, in fact, by this agonist and antagonistic axis. So in this case, a shift could enhance the signal, so can make it more stronger. On the other hand, an inhibitor or agonist can also make the compound less toxic. So that's the principle. This is again the roadmap, and this should show where we can plug in by specific causal adverse assay evaluation. So, and Monika, she will show you an example for the reactivity assay where she is blocking the where she's enhancing, in fact, the sensitivity of a compound in terms of cytotoxicity, and she will also show the effect on inflammation, how that happens under the LPS conditions under 3D conditions. So there are a lot of other possibilities where causality assays can be applied. And I just want to hand over to Monika and just want to point out here, this is using the ATP assay, but modifying the pathways which are potentially being involved in a DILI mechanism and making the decision whether this is relevant or not relevant for the DILI mechanism. So I'm now handing over to Monika.
Let me briefly guide you through two examples of causality assays that can be performed in InSphero's 3D human liver microtissues. The first one describes the causal relationship between reactive metabolite or ROS formation and glutathione. As you know, adverse effects linked to reactive metabolite or ROS are recognized as one of the key reasons for marked drug withdrawal or drug-restricted use. Therefore, the sooner the risk of these adverse effects can be assessed, the higher the chances of bringing a safe drug to the market. In cells, the burden of reactive metabolite and ROS is handled by glutathione pool. Here, it is noted as GSH. Glutathione both scavenges diverse oxidants as well as conjugates to and deactivates reactive intermediates. This is also happening in microtissues, cultured in standard conditions, where the pool of glutathione is sufficient to perform its detoxification role. However, if BSO, butyronine sulfoxamine, is introduced to the culture, the synthesis of GSH is inhibited, leading to depletion of the pool of glutathione and compromised detoxification capacity of the cells. This is represented in the left graph, indicating the changes in the levels of glutathione in standard, here shown as minus BSO conditions, and upon modulation with BSO. The experimental conditions presented here were carefully selected not only to ensure stable depletion of GSH from the cells, but also not to affect cellular viability, here assessed by the level of cellular ATP, as demonstrated on the right graph. As a side note, we would like to stress that careful design of the experimental conditions is critical to ensure both assay sensitivity and specificity. In other words, one needs to find the optimal conditions for a sensitization of the in vitro model to a given stimulus like BSO, but not to over-challenge the model and not to perturb other parameters that you eventually want to assess in your final investigation. So now, with these established experimental conditions, let's see the outcome of testing the classic model compound acetaminophen. As you know, acetaminophen is one of the most famous over-the-counter analgesics, but at the same time, it's also one of the major reasons for drug-related death due to liver toxicity, especially upon overdose. For the sake of time, we will not go into details of biotransformation of acetaminophen. However, let me just mention that reactive intermediate, NAPQA, when produced in small quantities, can be effectively scavenged by GSH. However, in the absence of GSH, and this is happening, for example, in case of overdose, but here in the in vitro model, upon exposure to BSO, very rapid cellular stress and damage can be any longer compromised, and that leads to a fast hepatotoxic effects. In the in vitro human liver microtissue model, the outcome of these events can be monitored by the decrease of cellular ATP and a very clear shift of the dose-response curve between minus and plus BSO conditions, as shown on the left graph. Additionally, one of the advantages of the model is that it allows for bundling several cytotoxicity markers on a single microtissue level. In this case, intracellular ATP measurement was bundled together with a release of cytoplasmic enzyme LDH. Both parameters were measured for a single microtissue, and as presented on the right graph, we observe a clear change in the release profile of LDH upon acetaminophen treatment in the presence of BSO. This indicates a membrane damage occurring in these specific experimental conditions. I think that one of the very frequent questions asked by people considering working with complex multicellular models, especially primary in vitro models, is its reproducibility. So, careful selection of cellular material and standardization of production is actually bread and butter for InSphero. And this further translates to assay reproducibility also in the space of causality assays. Here, for example, we see changes of cytotoxic response to acetaminophen in standard and glutathione depleted conditions, so the plus BSO conditions. This experiment was performed across 3 separate productions of 3 individual co-culture models where NPC lot was changed at every production. And I hope we can all appreciate the outcome of this comparison. So now I would like to move to the second causality assay to be discussed today. It's the causality assay investigating the risk of idiosyncratic toxicity linked to inflammation. Specifically, we're looking at innate inflammatory component that is stimulated in the in vitro model with LPS. This stimulation of cells, especially Kupffer cells, leads to release of inflammatory cytokines and chemokines, such as IL-6 and TNF-alpha. This application pulls out yet another feature of the model, its modularity. So what does it mean? This means that the microtissues can be built either as a co-culture of hepatocytes with other cell types, such as Kupffer cells and liver endothelial cells, or more simply as a hepatocyte monoculture, obviously lacking the immune component. This combination of these 2 models with and without LPS treatment can precisely illustrate the link between cytotoxic outcome and inflammatory component. And we would like to demonstrate this application with an example of 2 well-known antibiotics. The infamous trovafloxacin that was eventually taken out of the market due to acute liver failure and cell death, and levofloxacin with no associated idiosyncratic adverse drug reactions. If you look at the data on the right-hand side, it is clear that the cytotoxicity profile of levofloxacin is shifted in the presence of Kupffer cells and upon LPS stimulation, and this is shown by both ATP and LDH markers. This proves the hypothesis of the risk of hepatotoxicity linked to inflammation, and for further confirmation, you can see that safe levofloxacin does not become more toxic upon either IPS stimulation or in the co-culture model. This brings us to the conclusion of the presentation, where we were discussing how 3D liver microtissues can be used for causality assays with specific pathway modulators. So this type of investigation employs high throughput, compatible and cost-effective endpoints, such as ATP and LDH, in combination with specific pathway modulators. Today, we just presented an example of 2 modulators, LPS and BSO, but the panel can be expanded based on the research question and, for example, include antioxidants, bile acids, other inflammatory components, or lipid loading, just to list a few. Of course, this type of investigation might be triggered for a specific case of the DILI mechanism. However, we think that another attractive application is the screening tool. As already mentioned before today, the model presents a lot of advantages, making it suitable for screening purposes. These include, for example, high model reproducibility and production standardization, possibility to work with customized donor material, and customization of model complexity. So we can work with a simple monoculture model of a co-culture with different cell types. We should also stress the importance of platform that the microtissues are cultured in. And these platforms, especially the Acura 96 and the Acura 384 plates, allow for very safe culturing, very precise dosing, sampling, and endpoint execution. And this is all done in a very scalable manner. So at the end of the day, by the choice of the model, together with the platform, the endpoints, and in case of the causality assays, also the pathway modulator, one can build up a recipe for a scalable mechanistic investigation. With this, I think we can close the presentation part of the webinar and move to the Q&A session. So we hope there were some questions submitted by the audience, and let's see how we can address them openly now during the webinar.